As the sun sets and darkness falls, a tightly coiled night-blooming flower bud rapidly unfurls its pristine white petals. This behavior follows a predictable schedule. This historical illustration of Linnaeus's floral clock maps specific hours on the inner ring to the opening of corresponding flowers on the outer ring. For most plants, reproduction depends on animal pollinators. During the day, they use saturated colors to signal these insects. But as the sun sets, that strategy fails. Visual cues like red and blue pigments are nearly impossible to see in the dark. To adapt, night-blooming plants developed pale petals to reflect the moonlight and concentrated sweet scents that act as sensory beacons for pollinators traveling through the night. It is a complex feat for a plant, which lacks both a brain and eyes, to activate its scent factories on such a precise schedule, rather than simply reacting to the fading light. In the 1960s, botanist Lillian Overland began testing the limits of this behavior. She placed night-blooming jasmine inside laboratory rooms where light and temperature were kept constant. Even without the signal of a sunset, the flowers maintained a strict 24-hour cycle, releasing their fragrance right on schedule. Overland discovered that even when petals were severed from the main plant, they kept the rhythm. The timing mechanism is an autonomous biological oscillator, an internal clock ticking away inside individual cells. This internal timer provides an evolutionary advantage. By keeping fragrance locked down during the day, the plant stays hidden from daytime herbivores. This timing ensures a precise mechanical fit with nocturnal pollinators like the hawk moth, which uncoils its proboscis to reach nectar deep inside the flower's tube. Restricted scent release is a calculated resource economy. The plant only expends the energy required for fragrance when its specialized pollinators are active. To find the genes behind this timer, scientists today look to the petunia, using it as a model to map the microscopic assembly line that builds these scents. This diagram shows the metabolic pathway for scent. The green ODO1 box acts as the master switch. Above it, the red LHY box is the circadian timekeeper. During the day, the solid red line shows LHY acting as a brake, preventing ODO1 from producing scent. As the day ends, LHY levels naturally drop. This releases the brake, allowing ODO1 to trigger a rapid increase in the production of volatile aromatic compounds. The fragrance of a midnight garden is the result of a genetic switch precisely releasing its hold at the optimal time. On an ecological scale, this system creates a synchronized dance between flowers and the insects that visit them. However, this relationship is vulnerable. If environmental shifts throw off these internal clocks, the timing between a plant and its pollinator can move out of sync. Identifying these genes gives scientists the ability to intervene. By understanding the LHY and ODO1 mechanism, we may be able to manually adjust a plant's scent schedule. The goal is the development of designer crops, plants that can be engineered to attract new pollinator populations if their traditional ones disappear. The scent of the night is a specialized system of biological coordination. Understanding these genetic clocks offers a way to keep crops synchronized with the natural world, even as traditional ecological schedules begin to shift.